Kurinji Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
Free bus for LKG to 8th standard.
Free, hygienic and nutritious food.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
Kurinji Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Students, now in this video, so we are discussing complex tissues. So already some more information we have discussed in complex tissues. So in previous video we have learned regarding xylem. No, so there I have promised to give the diagrams on tracheids and vessels in this video. No, so that's why first of all initially I have drawn two diagrams of tracheids and vessels. Tracheid and one more is vessels. Next one in remaining two diagrams, no need to draw, but tracheids and vessels structurally you try to learn. So this is a structure regarding tracheids. So there you can see pit fields, yellow color within that. Empty space is there. No? Within the empty space or the lumen we can find out the which one? Pits, simple pits. Next one here, this particular cell wall is made up of lignin. Why? Because here all these cells are dead. No, dead cells means definitely cells or the cell walls are made up of lignin. Next one, vessels also, that will be like that only. So here elongated cells. So here the vessels are formed from the group of cells. So we will be taking as vessel members. So in yesterday's information we see now. So there also we can find out small pit like pit fields. So here, so all these ways, so these are all pit fields. So these two are tracheids and vessels. Next one, in previous video we have learned the information of exarch and like endarch condition. There exarch condition and endarch condition means once you see the exarch condition, so definitely we can see in roots. Best example. So there what is the information? Once you see here in the towards the center, no, in the center exactly you see its spotlight. So this particular part we will be calling as pith or medulla. In anatomy I will give you detailed explanation. So towards the center, which one is there? This perusa irukudana, this metaxylem. Chinna round we will be taking as protoxylem. This particular protoxylem is situated towards the outer side. Periphery we learned no in yesterday's class. Periphery means towards the outside. Towards the outside, which one is there? Pericycle. Towards the outside, here we are observing protoxylem. Towards the outside, periphery we see. Next one, inside we are observing the presence of metaxylem. So based on in this condition, no, so protoxylem which is situated towards the peripheral region now, outside. So that's why here this particular condition of xylem we will be taking as exarch condition. Exarch condition. Next one, like endarch condition. Once you see that endarch condition, exclusively we will observe in stems. In stems, say there. Here this is a small line is there now here. It is epidermis. Now towards the epidermis, which one we are observing? Presence of metaxylem. And to inside, towards the inside we are observing the situation of protoxylem. This kind of condition we will be calling as endarch. Endarch. Exarch means protoxylem which is situated towards the outer regions. That's why here we will be taking as exarch. and end arc means here the protoxylem which is situated towards the center. End arc condition we will observe in stem, and exarch condition we will observe in root. Suppose if you are unable to understand, sorry, what is exarch and what is end arc, we are unable to recognize the sir, means no worry, within 2 or 3 videos definitely I will explain. So here while explanation of which one, anatomy of root and anatomy of stem, in that particular case I will focus more on this arrangement of xylem only, no worry. Okay, by getting of confusion or the we will not get anything. Now this is a structure of a phloem. So in phloem, once you see their phloem there, our knowledge point of view 4 parts are there now. What are those? Like sieve tube elements, next one companion cells, phloem parenchyma, phloem fibers likewise. Now in this diagram we have drawn, we drawn how many number of structures, no? So 3 labelings we have drawn. Xylem phloem fibers are absent here. In this diagram we are not drawing phloem fibers. So what is the diagram they have given in our NCERT? So such kind of diagram only, so that diagram only here I have drawn. Now you see there, the first cell is sieve tube element. Once you see there, this particular one is sieve tube element. Now the edges of a sieve tube element, once you see there the edges of the sieve tube elements, so what we can find out, sieve plate we are seeing. Sieve plate. Next one, so here we are observing sieve pore. Okay, now here you see there, if you see this structure now, this particular first cell is there now, blue color. It is sieve tube elements. In sieve tube elements, one point we don't know, in a matured sieve tube elements, peripheral cytoplasm is there. Suppose if this cell, cytoplasm is pushed towards the edges, ends, so such kind of presence of cytoplasm we will be calling as peripheral cytoplasm, and nucleus is absent. That's why here we are not drawing presence of nucleus. And pakatali ipo rose color color is there now, cells. This cells name companion cells. This inge paranga companion cells. What is the use of these companion cells? Definitely attached with the or associated with the sieve tube elements. So here all functions of the sieve tube elements will be under the control of which one? Companion cells. Why? Because here the nucleus is there now, so that's why. Next one, here middle cell, same cell, the sieve tube element, so that's why here peripheral cytoplasm we are observing, and companion cells ipo paringa inge green color cells are there now, inge one, all these are all phloem parenchyma. இப்போ பாருங்க phloem parenchyma இருக்கு, companion cell இருக்கு, and one more is sieve tube element also is there. Means out of 4 structures of phloem, so 3 we have drawn. Now next one, which one we have neglected? Phloem fibers, so no need to draw here. According to the textbook information, diagrammatically this is the information. And one more point, once you see here, inge பாருங்க சின்ன hole like structures are there now, சின்ன pit fields. இது என்னன்னா hole like structures we will know. The hole like structures which joins, which associates the sieve tube elements which associates the which one sieve tube elements and companion cells. இப்போ பாருங்க, suppose here no gap means how these cells will be associated together. So that's why here gaps are there. These gaps only our subject point of view we are calling as pit fields. Okay, shall we start information to learn? Right, now companion cells information, this pink diagrams are there, no? This companion cells. So here what is information? No, these companion cells are specialized. Parenchymatous cells which are closely associated with the sieve tube elements. One more time, once you see there, companion cells or specialized parenchymatous cells which are closely associated with the sieve tube elements. Ipo paringa, idi which one, like sieve tube element, and this one is companion cells. If both are associated together or connected together because of the presence of pit fields. between their common longitudinal walls. So here the longitudinal longitudinal walls are there. There these two cells are connected together. What is the main important point? No, these companion cells helps in the maintenance of pressure gradient in the sieve tube elements. So this particular point you don't forget. NEET examination point of view important. So what is the point? So definitely these companion cells helps in the maintenance or the maintaining of pressure gradient in the sieve tube elements. So here because of the absence of nucleus in the sieve tube elements, so what is the pressure gradient is present in the sieve tube elements? No, is under the control of companion cells. Is under the control of companion cells. Next one, the phloem parenchyma. If you next one so first one we have seen sieve tube elements we seen. Next one now we completed companion cells, and third one is phloem parenchyma. One point you let me get from you, what is the companion cells closely associated with sieve tube elements? How companion cells are associated with the sieve tube elements? Once you say, because of the presence of pit fields, hole like structures are there, no, these are all. And main important point, what is the point of companion cells we are observing? So it maintenance, you see here last line, maintenance the pressure gradient in the sieve tube elements. Means what is the pressure gradient we will be or we are observing in sieve tube elements? No, that pressure gradient is under the maintenance of companion cells. Okay, next one, phloem parenchyma. Made up of, means this particular phloem parenchyma made up of elongated tapering cylindrical cells which have dense cytoplasm and nucleus. Cells how it will be elongated, enlarged cells like. Next one, tapering cylindrical cells. Tapering edges, cylindrical cells over there, and with dense cytoplasm, woolly cytoplasm also, very thick cytoplasm is present, and nucleus also is present. Next one, the cell wall is composed of cellulose and has pits. So here this particular phloem parenchyma is living only, no. So that's why author mentioned, so he was given information regarding the cell wall composition. Cell wall is made up of cellulose and has pits. Means definitely here what we can find out, like pits we can find out holes. And through which plasmodesmata connections exist. So means here once I have given bridge like structures which are present between the two cells for the transport of water and minerals. Likewise did I give? So that particular point, so is nothing but bridge like structures which are present between the which are present between the two cells. So those bridges we will be calling as plasmodesmata connections. All these are all connected together because of the presence of pits. Okay, next one, phloem parenchyma stores. Now what is the main important function of this particular phloem parenchyma? Means helps in storage, of food materials and other substances like resins, latex, mucilage. So phloem parenchyma main important points, cell wall is made up of cellulose. Next one, pits are there, no? All these pits are get connected with the Pachythalli cells because of the presence of plasmodesmata connections. Next one, what is the main important function of phloem parenchyma? Definitely helps in the storage of food materials. Not only food materials, definitely helps in the storage of resins, latex, mucilage. So in biomolecules you will learn one word, but now only I will give you, try to remember, that is secondary metabolites. Resins, latex, mucilage. So all these are all like waste materials or the waste products of plants, but which are useful to the human activities. Latex or some do you know suppose if you plant fruit, if you take the unripened one, if you pinch that particular support of fruit, milk will come, no. So that is actually it is excretory product to the plant, but it is useful to us. Likewise rubber plant, do you know, so that from the rubber plant we can take the rubber actually which is not useful to the plant, but by using that one, so we can prepare the rubber. eraser we will use, no, that is the thing. So likewise here all these kind of materials or the products we can be or storage process takes place in the phloem parenchyma. Next one, ipo idu neat bitama. The phloem parenchyma is absent in most of the monocotyledons. So while watching this particular line now, try to put your mobile in a pass mode and video and try to say this one 5 to 6 times. Okay, so definitely very important. Phloem parenchyma is absent in most of the monocotyledons. Phloem parenchyma is absent in most of the monocotyledons. So this 1 or 2 points related to the phloem parenchyma. Next one, phloem fibers point of view. So till now we have seen the information of which one, living tissues or the living cells in the phloem. Means our knowledge point of view, in phloem members 4 types of tissues are there, no, 4 members are there, or the phloem members we can be divided into 4 types there. 3 are living and 1 only is dead one. So that dead one only is nothing but phloem fiber. So this particular phloem fibers point of view, these phloem fibers we will be calling by one more name, important again, bast fibers. Bast fibers are nothing but phloem fibers. Ipu I told you one point, no, what is the point I told you? So here these phloem fibers are dead, I said no. Dead means definitely cell wall is made up of lignin. Adi we have seen in like sclerenchyma and xylem members also we see now, but in phloem fibers, even though so here these are phloem fibers made up of sclerenchymatous cells. Sclerenchymatous cells. Point you understood, ma? So phloem fibers also known as bast fibers and are made up of sclerenchymatous cells. Made up of with sclerenchymatous cells, and these are generally absent in, means here the fibers are absent in where primary phloem. So these are all absent in primary phloem. Suppose what is the point? Phloem fibers or bast fibers made up of sclerenchymatous cells. My dear students, don't forget sclerenchymatous cells. So next one here, ipo paringa. So till now we have been discussed. Regarding which one like C-tube elements we seen, companion cells we seen. Next one phloem parenchyma we see. Next one more is phloem fibers we seen. Now let us start to discuss regarding like 5 fibers only, last and final points. Let us see. So now here you see there, these are all absent. These are all means which one? Phloem fibers are absent in primary phloem. Phloem just now only it is a formed now. So there which one is absent? Phloem fibers are absent, but phloem fibers are found in or present in secondary phloem. This is important point. What are the fibers are there now? Phloem fibers. These phloem fibers are absent in primary phloem, but these phloem fibers are present in secondary phloem. Next one, these are much elongated. So now author given information regarding the phloem fibers. Now these fibers are elongated, unbranched. So definitely fibers will not be like a branched one. 100% will be unbranched and have pointed needle like structures. Pointed means very sharp arc. Okay, so very pointed and needle like apices and terminal part. If you say safety pin or needle, did you observe? So if you take a needle, like a stitching purpose we will use, so that one how much it is pointed. So very like a sharp pointed structure will be there, no? So likewise here also phloem fibers or needle like apices, rombo pointed structure will be there. And the cell walls of these phloem fibers is quite thick, very thick and dark. So why? Because here these cells are made up of sclerenchymatous, no? So that's why here the phloem fibers are quite thick. Up to here. Next one, at maturity, these fibers lose their protoplasm and become dead. Here it is important. We have seen phloem fibers are dead, we seen. When we are observing, suppose this particular dead condition, we are observing only when the fibers attains maturity. means when these fibers are in young stage, at the time what we can observe within the cells, we can observe the presence of protoplasm. We can observe when the particular fibers are in a young stage, but when fibers reach to the like a maturity stage, at the time only fibers lost their protoplasm and become dead. You need to try to save this point in your mind. Means only in a young stage particular phloem fibers maintains the protoplasm. When the phloem fibers attains maturity, at the time lost their protoplasm and become dead. Next one, phloem fibers of jute, flax, hemp and are commercially used. So means here jute, flax, hemp and are commercially used. Already in the previous video I have given information regarding this one. So their jute or their flax is there, hemp is there. Next one, there are different types of flaxes or the fibers are there. All these are all commercially helpful. So up to here, so we can study the information regarding phloem fibers. Next one, so next one here information extinction related to the phloem. The first formed primary phloem consists of narrow sieve tubes. So if I say all points are important, you will not watch videos, that's why I will not say that word, right? But important, try to watch, right? So first formed primary phloem consists of narrow sieve tubes. First primary phloem means first formed one. First formed phloem is there means there very sieve tube elements are there. But these sieve tube elements so sieve tube elements how with narrow sieve tube elements are there. Means here very chinna like compressed sieve tube will be there. Next one, it is refers to as protophlloem. Next one, later formed phloem has bigger sieve tube elements or the bigger sieve tubes. So means first formed one their sieve tube elements are not much developed, but later formed phloem is there. Their sieve tubes are bigger one. This particular one only we will be taking as metaphloem. Simple primary phloem is there no. Primary phloem, our knowledge point of view, we can be divided into two types. One is protophloem and the second one is metaphloem. In previous video we seen primary xylem is there, means that primary xylem we have been divided into protoxylem and metaxylem. Likewise here also primary phloem is divided into protophloem and metaphloem. Protophloem nalli narrow sieve tube elements are there, and in metaphloem nalli what we are observing bigger or bigger sieve tubes we are observing. That is information related to the phloem. Okay. Next one new concept that is name is or the side heading is tissue system or this tissue system. What is the main important points? Based on the or on the basis of their structure, location and their so means one more time. Tissue system is there this particular tissue system only based on their structure and based on the location. So three types of tissue systems are there. How many types are there? Three types. So there first one is epidermal tissue system. First one is epidermal tissue system. Second one is ground or fundamental tissue system. Third one is vascular or conducting tissue system. Once you say along with me, tissue system, on the basis of the structure and the location, tissue systems are divided into three types. So first one is epidermal tissue system, second one is ground tissue system or fundamental tissue system, and third one is vascular tissue system or conducting tissue system. Now let us start to discuss first one in tissue system. First one is epidermal tissue system. Epidermal tissue system. Now here epidermal tissue system forms the outermost covering of the whole plant body, comprises up to here. Let us see. Epidermal tissue system forms the outermost covering layer of the whole body. means what is the point? Outermost protecting layer is nothing but epidermal tissue system. Ipo suppose outside is there means that one only must play an important role in terms of protection, isn't it? Suppose inner one is there means that one will do another function, but suppose if any kind of tissue system is situated outer side means definitely that particular function we can guess definitely helps in protection. Emma. So and comprises, so what are the cells that generally are present here means epidermal cells are there, stomatal cells are there, and few epidermal appendages are there. Appendages means outgrowths. Okay, appendages are there. What are those? Trichomes and hairs. Trichomes we will observe in stem, and root hairs or the epidermal hairs we can observe in leaf and roots. Okay, right. Now epidermis is the outermost layer of the primary plant body. Already we have seen it is made up of, means here how this particular epidermis is made up of means it is made up of elongated compactly arranged cells which forms a continuous layer. Appadina what is the main important point? One more point to be discussed. When the cells are elongated and compactly arranged cells, appadina, so based on this one what is the point here we have to understand now? Between the cells there is no intercellular spaces, so intercellular spaces are absent. Elongated cells are there, compactly arranged cells are there, which forms a continuous layer. Appadina, between the cells there is no gap. Suppose if between the cells a gap is there means that particular gap our subject point of view we are calling as inter. Inter means between the cells only, no. That's why intercellular spaces are present means intercellular spaces are present we will write. Suppose between the cells there is no gap means, so how we will write? Intercellular spaces are absent. Am I getting only? No. Likewise, once you see the cells are elongated compactly arranged cells which forms a continuous layer. For continuous layer we can observe. Next one, epidermis is usually single layered epidermis we will observe. Appadina ore layer protecting layer will be there. Maximum cases no epidermal tissue is single layered maximum cases, but nerium therima in a highway like a pink color flowers. Did you see plants? So that particular plant leaf no, that one multi layered అది మల్టీ లేయర్డ్ ఎపిడెర్మిస్ ఇరుకన అది xerophytic characters means the plants which will grow in desert areas no, so ange so like tani availability will be very less no, so that kind of character we will observe in nerium. So that one you leave it, not required now. So definitely right. Now epidermal cells or parenchymatous cells. Parenchymatous means munadi we see no, simple tissues are divided into three types: parenchymatous, cholenchymatous, sclerenchymatous. Likewise we see no, now these epidermal cells are made up of parenchymatous with a small amount of cytoplasm. Dense cytoplasm illama konjam mattunda irukku. Then dense cytoplasm is not there, small amount of cytoplasm mattunda irukku with lining cell wall, cytoplasm lining cell wall, and a large vacuole is there. Here large vacuole is there and koncham cytoplasm is there, but here one point they didn't give in a textbook, but I will give you what is that point. No, in epidermal cells no, even you take leaf also. Even if you take epidermal cells of leaf no, there chloroplast is absent. Maximum suppose if you take leaf means are there chloroplast is present no, likewise we will think. But even though when you will observe the epidermis of leaf no, there we could not see one thing or we could not see the presence of the chloroplast. So thereby here one point you try to say the epidermal cells of leaf not possess chloroplast, but the same chloroplast we can observe in where is stomata? We can observe in where the stomata? We can observe in stomata. Here you see there. In stomata again we can see the presence of chloroplast, but in epidermal cells no, chloroplast is absent. This is a neat bit try to save in your mind. Okay, next one. up to here completed. Now vacuole is present and a small amount of cytoplasm and a lining cell wall also is present. Next one, outer side of the epidermis is often covered with a waxy thick layer is known as cuticle. Ipo outer side of the epidermis means if you see there the leaf suppose for example if you see there no upper epidermis leaf, ipo if you take a dicotyledonous leaf upper side no one color will be there, kele one more color will be there no. So reason I have given in previous videos. Now what is the point? On the outer side of the epidermis, so we will observe a waxy thick layer. Ipo do you know like a nilambo lotus flower leaf, did you observe? Suppose if you keep one drop of water, if you keep there no that particular one drop of water like it will comes very close and it will be seems to be like a mercury. Emma, did you observe the like lotus plant leaf? So likewise there no on the leaf what we can observe no waxy thick layer. That particular layer only our subjective point of view we are calling as cuticle, okay, which prevents the loss of water. So when it is present, when it is situated on the upper epidermis no definitely helps in the reducing of loss of water. Reduce the loss of water only we will be calling as or it helps in reducing of water loss. Next one, the cuticle is absent in the roots. Ipo roots generally are present in within the soil no. Sir, ipo roots even though present within the soil, cuticle irika sir epidermis male means no ma, no chance to the presence of cuticle in the roots. Okay, this particular point also try to save in your mind. Next one, stomata or the structures present in the epidermis of leaves. Now only we see no stomata means some kind of specialized cells, so which are present in the epidermis of leaves helps in transpiration and losses of water, and one more is these stomata members helps in exchange of gases. Two important functions are there related to the stomata. One here I didn't write, but through my voice you can get. What are the functions of stomata? No, one is exchange of gases and one more is helps in transpiration and a loss of water. Okay, that particular loss of water also helps for the taking of water and minerals from the soil. How it will helps and everything. So in the next chapter I will give you explanation. So in this video, so what are the main important points we have seen? No, we have continued to discuss the phloem information. So later we have started to discuss about the tissue system. In a tissue system, epidermal tissue system, very fewer points only, somewhat information only we have discussed. So in the next video I will give you continued information of epidermal tissue system. So watch these videos and get the knowledge and be safe at home. Thank you.